All right. Last time we left off talking about the level of regulating genes expression at the mRNA level. So we talked about chromatin level, we talked about transcriptional level, and now we're talking, okay, you've actually decondensed your chromosome enough, and you've got RNA polymerase down, and you've made an mRNA, the cell still can regulate whether or not that gets turned into a functional protein. All right. So we started talking about alternative splicing, and I'm not going to go into that anymore. Suffice it to say, the cell can alternatively splice transcripts, so you could make several different mRNAs off of the same gene region. And we talked about way back the fact that the cell can actually alternatively edit the mRNA. So you can splice it in certain ways, and then you can also change nucleotides. And so whatever's in your the loci actually in the genome, the cell is able to edit that at the mRNA level, and you might actually be able to change which proteins you code for by changing nucleotides in the mRNA. I want to talk now about non-coding RNA, because there's an additional level of control here. You can decide whether an mRNA gets alternatively spliced or edited, but you can also then determine whether or not that mRNA actually gets translated into protein. And you can do three things. You can either hold off and keep that mRNA around and not make it into protein. Or you could degrade that mRNA and completely remove it, right? We talked about scrambling the jets, right? You get an mRNA made, but then you might just degrade it immediately. A third thing you could do is you might actually amplify that. You might additionally make more copies of the mRNA. So you can amplify, you can pause, or you can stop, right? A lot of this is done by non-coding RNA. So 1.5% of the genome is actually translated, human genome is actually translated into proteins. So if we just surveyed the entire genome, the 3 billion base pairs, only 1.5% of the nucleotides are actually involved in coding for amino acids and coding for proteins. Right? The rest of it is doing something else. And for a long time, we thought it was junk DNA, right? And we've talked about this before. But 90% of it, or more maybe, the number keeps increasing as we learn more about this, of the genome is transcribed. That is, it's made into some type of RNA, but not translated, right? So some of this we already had known about for a long time, tRNA, ribosomal RNA. But there's also these non-coding genes, non-coding RNAs, and there's whole separate classes of them. There's miRNA, that stands for microRNA. There is snRNA, that's small nuclear RNA. SNO, SNO RNA, that's small nucleolar. So not only in the nucleus, but it's specific to the nucleolus. There's pRNA. There's long non-coding RNA. I'm going to focus on our talk on these guys called microRNAs. And I'm going to talk about how they're involved in regulating mRNA transcripts. The actual transcripts that are going to get made into protein are going to be regulated by the interaction they have with these small microRNAs. Okay. We know in the human genome at least 700 microRNAs exist. And that's an old number too. That's a number I found actually from last year. I haven't looked it up yet, but again, this keeps growing because we keep finding more and more of these. So at least 700 microRNAs. We've found locations, 700 different locations in the genome where microRNAs are made. And at least 10,000, this is also an old number, at least 10,000 protein coding genes are estimated to be regulated by these microRNAs. So 700 at least microRNAs are regulating 10,000 or more actually protein coding genes, mRNAs, right? You have about 25,000 protein coding genes. At least 10,000 of them are known to be regulated at the mRNA level by these microRNAs. Okay. We've also identified a bunch of what had previously been called pseudogenes. There are regions of the genome that look like they should code for protein, but they've got like a premature stop codon in them. So if you're looking through the genome and you see what looks like a gene, it's like got seems like protein coding sequence, seems like it's got a promoter to it, but then you actually look at the sequence and there's a premature stop codon. That had been called a pseudogene. Looks like a gene, but it's probably not going to get made because it's going to have this premature stop codon. The more we learn about microRNAs, though, and the ability for mRNA editing, those pseudogenes have kind of become zombie genes, which is what was thought to be a dead gene actually can be edited and made into a functional protein, right? So it comes back alive out of the grave, right? So they've called them zombie genes because they may actually have function. So there may actually be more protein coding genes than we originally thought, because these might actually, even though they look dead, they might actually rise again. So I'm going to talk about these micro or small RNAs and how they do this regulation. There's actually two classes of these small RNAs. Small interfering RNA called siRNA, and then there's microRNA, miRNA. There's subtle differences in the way these things are formed. I'm just going to lump them together. I don't think the differences are very significant, so I'm going to give you kind of the basic idea. But what they do is they come from precursor RNAs that are made off of the genome, right? So these are actually, in a sense, they're genes, right? They're locations in the genome that get made into RNA and provide a function. So the term gene has been expanding. We used to think of a gene as just a thing that coded for protein, but now these, in essence, can be considered a gene, right? They're a location in the genome that does something. They're double-stranded to begin with, and polymerase, RNA polymerase, comes and sits down and makes a copy, just like an mRNA would get made, right? So it's transcriptional. But they have a repetitive region in them that causes them to make these stem loop structures. So here is RNA polymerase is making this double-stranded RNA. It makes a single-stranded piece of RNA, but that folds back onto itself in these stem loop structures, right? They have internal complementary sequence. So it's a single strand that gets made by the polymerase, but that single strand base pairs to itself and makes these stem loop structures, okay? Those stem loops get cut by this protein called dicer. So this is just a normal protein coding gene. And what dicer does is it grabs this double-stranded RNA and it dices it up. It actually cleaves it like a restriction enzyme would cleave DNA, and it cleaves it into these smaller pieces, okay? This is when it becomes micro or small interfering RNA. So it starts off as a bigger, longer piece of this double, stem loop double strand, and it gets diced up into these smaller ones. These smaller ones that get associated with a bigger protein complex called the RISC, which is the RNA-induced silencing complex. Okay. Now, it's the RISC is what is going to determine one of the three things that we do with the mRNA, whether we amplify the message, whether we stall and just pause the message from being made into protein, or whether we actually go and degrade it and just stop that gene's expression, okay? We're still trying to figure out how this works, but we at least know these three distinct pathways. What the small RNAs do is they basically are acting like the target or the probe. They have complementary sequence to the mRNA that they're going to regulate. So the RISC is going to be the thing that determines whether the mRNA that we're regulating gets amplified, paused, or degraded. It's the small little RNA piece that acts as the specific target to say, like, this is the mRNA that we should bind to and do one of those three functions to it, right? So these small guys have sequence specificity to mRNAs. And so that's what's going to bring the RISC complex to the mRNA that we're trying to regulate. These can come from multiple places in the genome. Sometimes they come from what we thought were pseudogenes, right? It looked like a duplicated gene, because it had similar sequence to a known protein, mRNA. But instead of being a pseudogene, just a non-functional thing, it actually just was coding for a little specific target sequence to go back and recognize the mRNA and regulate it. Sometimes these are actually found in the gene itself. So the gene might actually get made into an mRNA, but then another subset of the gene actually just gets made into the microRNA so that it can regulate itself, right? So the origin of the microRNA is the same locus, the same location on the genome as the mRNA came from. Now it would be the opposite reverse strand that it comes off of, but these can come from multiple places. So then the RISC complex can do one of three things. It can either, so I've got my little double-stranded, this is this little appendage here on the RISC, is this double-stranded micro or small interfering RNA. One of the strands gets removed, so I've got a single strand, and that single strand then is acting as my like antisense probe to go find the mRNA that's appropriate. Okay, so here is an mRNA that it's bound to. Okay, so I've got a little my little probe sequence. It finds the mRNA, and in this case, it's labeled M7G. I don't know why. It's from whatever review paper I pulled this out of. But this is a target mRNA that it's found, right? What this can actually do is recruit in more RNA polymerase and might actually make you copies of that mRNA. So the RISC complex might actually amplify the number of mRNA strands that you have. So it's going to amplify this gene's expression, right? Turn on more mRNAs, right? Make you more copies of the mRNA, and so you actually produce more protein than you would have, right? So the RISC complex can amplify, or it might bind, and so the little probe finds the mRNA, but the RISC might actually at that point bring in RNases, enzymes that chew away and just degrade the mRNA. Right, so that would be the degradation pathway. Or the RISC complex might use its little probe, find the mRNA, but then it just sits there and just blocks the ribosome from being able to grab it. So this is the pausing mechanism, right? It might not amplify, it might not degrade, but it might just sit there and wait until the appropriate time for that protein to be made, and then the RISC would dissociate from the mRNA and let the ribosome continue to make the protein. So I put up this generic cartoon because I don't want you to get bogged down in the details, and we're still trying to figure out the details of this. Suffice it to say that we don't quite know exactly what makes up the RISC, right? There's going to have to be some difference in the RISC in order to recruit things to amplify the mRNA. There's got to be certain proteins that are different that are associated with the RISC that cause degradation. There's got to be different proteins associated with it in order to just be the pausing mechanism. So in different cells and in different times and in different organisms, we're still trying to figure out what those proteins are and what the differences are and how the cell codes for that. But I just want you to recognize that there are these three possibilities that the mRNA could be doing, right? So it's this small microRNA and the RISC that can do one of these three things depending on the cell's context, right? So
the RNA can have one of three fates. Why would the RNA, does it get made regardless and then it's decided what is to happen to it? It seems, I just was thinking, why would you make the RNA just to have it degraded?
Yeah, well, I mean, honestly, we don't know. The best conjecture I can make is kind of this, scrambling the jets kind of thing, right? You might need it, you might not, and so if you don't need it, degrade it.
Oh, I see.
Right. If you know you need it, but just not yet, then you would just pause it, right? But you might not need it, so you just degrade and make some more, degrade and make some more. It seems wasteful, but in a sense, if what you're after is really specific timing of your gene expression, then it's worth it to be wasteful a little bit to make sure that you've got an mRNA when you need it. But if you don't, well, you just degrade it, right? So there's like a trade-off of, do I waste energy or do I risk being unprepared to make the protein I need, right? So.
I'm wondering when the double-stranded RNA is first transcribed,
It's single-stranded. Yeah, it's a big, long piece of single-stranded RNA, and then it folds back onto itself to be a double-stranded thing. And then Dicer grabs those. So this is the stem loop. The little stem got trimmed off, and so I've just got double-stranded RNA because it's self-complementary. And then the Dicer comes in and slices that longer double-stranded RNA thing into the small pieces. Right? Then those small pieces get associated with the RISC, and one of the complements gets degraded, so I'm just a single-stranded thing. So now I'm a single-stranded RNA that can go find complementary mRNA to regulate, right? So what the small RNAs are is they're just a mechanism to know which mRNAs to go bind to, right? They provide the specificity by base pairing to an mRNA that says that's the mRNA we're going to exert our function on. And it could have been any one of these three functions, but does that make sense?
Yes.
Okay.
When the first double-stranded RNA,
Yeah.
when it folds on itself, how come it doesn't form loops?
It does form loops.
Oh, okay. So
And sometimes those are cleaved off, and we're still figuring out how Dicer works too. And so yeah, it does. Here, this illustration is showing you perfect base pair complementation. It's not really that case. There are loops in places where it's not. But then Dicer comes and slices that up, and you might actually lose that little loop that used to be there. Might not be a part of the actual small RNAs that you need. Right? Sarah, and then Brian.
I think you already answered. I was just confused about where microRNA came into the picture and small RNA came into the picture.
Yeah, small inhibiting and micro, I'm using those terms interchangeable. There's subtle differences in the way they're made and what their size are and stuff. But basically, they're both processed through Dicer. And so you make a long original transcript that wraps on itself to become double-stranded, and then you slice that up. Dicer slices that up into really small, they're only something like 19 to 21 base pairs, very small little pieces that are actually providing the sequence specificity to go find the appropriate mRNA to regulate. Yeah.
Could that sequence specificity be because the stem loops are made from the actual gene itself?
Right. Sometimes they're made from the actual gene, right? And so that's what gives you the sequence specificity. Sometimes there's, complementary or identical sequence of the mRNA found someplace else in the genome, and that gets made into the microRNA, and so the complementary sequence can find the other one. Right. So you have to have duplication of the sequence somehow, either by making two copies from the same location or having those two sequences other places in the genome. Yeah. Other questions about micro? Natalie looks confused. No? Okay, you're good. It's just me. I'm misreading your facial expressions. All right. The last point of regulation is to actually get a protein made, right? Proteins are not necessarily functional. They're not actually going to go do their function just because you folded them properly and got them made, right? Proteins can toggle back and forth typically between an on and an off state. So you might have a protein, but it might just be floating around either in the cytoplasm or in the membrane or in the nucleus, wherever it is. And it might be there but not functioning, might not be turned on. One of the common switches is phosphorylation. Sometimes you have an unphosphorylated protein. If you phosphorylate it at a certain place, it'll turn it on. Sometimes phosphorylating it turns it off. So it's not a strict rule that a phosphorylated protein is always on. It depends on the cell context and the protein context. So sometimes it's phosphorylated and that's turned off, and then removing the phosphate turns it on. So just know the switch could go either way. It's like one of those confusing switches in your kitchen where there's two switches for the same light. And so sometimes turning it on turns it off, and sometimes putting it down turns it on. It could be either way, right? The other thing is, I don't even know what this illustration is showing. You can also target proteins to be degraded, right? So you can toggle a protein on and off, right? You might need the protein's function, but not right now. So you just keep it in the off state. When you need the protein, you turn it on. If you don't need it, you turn it back off. You can toggle back and forth. Sometimes the cell decides, though, I don't ever need this protein. I don't need this protein anymore at all. Right? There's no reason to keep it around. So you actually target the protein for degradation. So one of the ways you can do this is through adding a molecule called ubiquitin. Ubiquitin is a little peptide molecule. You stick this peptide ubiquitin on a protein, and that targets it to be degraded. Proteases are enzymes that degrade proteins, and there's a complex of proteases called the proteasome. Basically, that's where proteins go to die, right? You tag it with some type of label that says, I'm done using this protein. Oftentimes, that's multiple little ubiquitin peptides. You stick these bunch of ubiquitin peptides, so I think that's what these are. A bunch of ubiquitin peptides on it. That targets it to go to this proteasome. The proteasome digests away the protein. And then you can reuse those amino acids and go make whatever protein that's appropriate to make now, right? I'm not going to go too much more into, this is much more complex than I'm giving you, right? I'm saying on and off or degraded or not. But if you want to know more about this, you can take biochemistry, metabolic biochemistry. We go into this a little bit more. But suffice it to say, you turn them on and off, or you can get rid of it completely. Right? Questions about protein regulation?
So if it was going to be turning it off, it would be like as a repressor kind of thing, like to repress some inactive something, inactivate something so something else can be active.
Yeah, well, it's like, I'm trying to think of an example to make it clearer. Your cells don't always metabolize glucose, right? Sometimes your cells metabolize glucose, sometimes they metabolize proteins, sometimes they metabolize fats. So the first enzyme in that step is hexokinase. It takes a glucose molecule and starts sending it down the glycolysis pathway. Well, your cells need that enzyme around. But if they're not metabolizing glucose immediately and they're using a different pathway, you don't want to degrade that protein because maybe 2 minutes from now you're going to want to metabolize some glucose, but not right now. So you can inactivate the protein, just like phosphorylate it, which turns it off. So the cell says, I'm not going to do glycolysis right now. I'm going to turn off those enzymes, and you turn on other enzymes to do the metabolism of fat right now. But then you want to turn back the glycolysis pathway on, so then you remove the phosphate and allow that pathway to go. So it allows the cell to toggle between different decisions to make. Now, if you're a kind of cell that just never metabolizes glucose, that's just not what you do, right? Are there any cells that do that? Probably not. That's a bad example. But certain ones. Well, every cell in your body is going to metabolize glucose at some point, right? So it's a bad example, but say fat metabolism. Not every cell in your body, like your brain cells. Your brain cells metabolize very little fat. In fact, maybe none at all, right? So they would just, if they ever made those proteins, they'd just degrade them, right? There's no reason to make those proteins. So at some point, a cell decided to become a brain cell, a neuron, and so it just shut down any proteins that were metabolizing fat, and it just decided to exclusively metabolize sugar, right? So at that point, when that cell made that decision, it would just degrade all of the proteins for fat metabolism and never make them again, right? So it's just a more permanent way to stop making that function, stop doing that function. No, it happens in development when cells are choosing to be, but mostly this is actually happening at different stages of a cell's life, right? So the liver cell, needs to metabolize some toxin that's in your body. So it expresses this protein. That protein, does whatever it needs to detoxify that substance. That substance isn't around anymore, so the proteins would just get degraded. And if I ever encounter that protein again, or that substance again, the liver cell could make the protein necessary. It'd do its function. But if that's a rare case, then you just degrade the protein because you don't need that function anymore. So this is just like the last step in regulation, right?
So when you phosphorylate, you're turning it off.
Not always. Some proteins, when you phosphorylate it turns them on. Some proteins, when you phosphorylate them, it turns them off. It's protein specific. So, All right, I want to back up and just for a couple of minutes. Oh, actually, do we have? Yeah, we've got 5 minutes. We're going to talk about a bigger picture of gene regulation. Sometimes it's the cell itself that's making the decision about whether or not to turn genes on or off, right? When we were talking about the lac operon, I mean, all we're talking about is a bacterial cell, right? And the bacterial cell itself changes its gene expression, right? If glucose is around or if lactose is better to do, the cell is signaling within itself. Mostly in your cells, though, your cells are changing their gene expression sometimes by their local environment and how they're responding to it, but oftentimes certain cells are signaling other cells to change their expression, right? Cell-cell signaling is happening a lot. So an adjacent cell might actually be telling another cell, you know, we should be doing a different function, and so they signal to it. So I want to talk about signal transduction. So a cell itself might not change its gene expression until it gets a signal from a neighboring cell. So one of the ways is through signals called ligands. If a signal is coming from a separate cell, oftentimes it's a secreted molecule, probably a protein. It's been secreted from an adjacent cell over here. It secretes this messenger, and the way it targets and tells the other cell to change its gene expression is first by bonding to a receptor. So this requires both the cells to have some specific, agreement about what they're going to do, right? In order for this cell to respond to that signal, it's got to be expressing this receptor, right? So cells must be already primed in a sense, like they know that they're the kind of cell that needs to sometimes receive that message, right? So this cell has to be expressing the receptor, putting that protein in this membrane, and then it's basically capacitated, right? It has the capacity now to respond to a signal if it in fact comes, right? So in this case, it's expressing the receptor, a signal comes, it actually bonds to the receptor, and that turns on in some way, activates what we call secondary messengers, right? So the primary messenger is the actual thing that the adjacent cell secreted. Inside the cell, when that receptor actually bonds to a signal, it's going to turn on other proteins. We call those second messengers. Because the idea is one cell is going to tell the other cell to change its gene expression. So the primary interaction is between the two cells. The secondary messenger is between the receptor and things in the nucleus to actually get genes turned on, right? Oftentimes, second messengers are proteins, and the receptor will, for instance, like phosphorylate a protein, right? So if a signal hits this receptor, that activates a kinase domain, and that kinase domain phosphorylates a second messenger. And then oftentimes a second messenger will phosphorylate another protein, and that one will phosphorylate another protein. You get this big cascade of, kind of playing telephone, right? This protein delivers a message to the receptor, and the receptor tells one second messenger, and that one phosphorylates another second messenger and phosphorylates another, so you can have this big, long cascade. Eventually, though, what happens is some protein must make it into the nucleus and activate a transcription factor, right? Because if we're going to get new genes to be expressed, genes get expressed because a transcription factor sits down on the genome and recruits in RNA polymerase to actually turn on a new gene, right? So at some point, these second messengers must enter the nucleus, activate a transcription factor. That transcription factor sits down, recruits RNA polymerase, and now we're expressing new genes in response to that environmental stimulus, that message from another cell. Okay. This is one mechanism of cells changing another cell's gene expression. The second example I want to give you is, it's easier. It's a simplified version of this. This is mediated by what are called nuclear hormone receptors. In this case, the primary messenger is a hormone, and it's usually a steroid hormone. Hormones are just signaling molecules. So you can have protein hormones. Proteins don't make it through cell membranes, right? So if you're a protein signal, you're probably not going to be able to make it through the membrane. You're going to have to bond to a receptor, okay? If the signaling molecule is a hormone, hormones usually are capable of penetrating the plasma membrane. They themselves will get into the nucleus. So here's the plasma membrane, here's the nuclear envelope. The steroid is going to be able to be secreted, pass through the membrane because it's going to be a lipid. Steroids are lipids. So it's able to hydrophobically get through. It's able to get into the nucleus. And then it actually binds to a receptor. In this case, the receptor is not on the membrane. The receptor is a protein inside the nucleus. So this is a nuclear hormone receptor, right? This guy receives the signal. The steroid hormone binds to this guy, and oftentimes it's that guy itself that is the transcription factor. So if he gets activated by a hormone, it will sit down on the DNA, recruit RNA polymerase in, and you'll start expressing new genes. This doesn't require that secondary messenger thing, right? The primary message gets all the way into the nucleus, and then I directly stimulate that transcription factor to turn on new genes expressing. So it's kind of like a shortcut. Questions about either of these two? They're kind of like bigger picture, right? Because all of the secondary steps are now in effect, right? Once you get, the chromosomes decondensed and you get a transcription factor here, then you can do mRNA level regulation and splicing and all that. Right? This is bigger picture. How do you start it in the first place?
Biola University offers a variety of biblically centered degree programs, ranging from business to ministry to the arts and sciences. Visit biola.edu to find out how Biola could make a difference in your life.